Hello, everybody, and welcome to this webinar. We're going to give people a few more minutes to join. I see lots of people are joining right now, so the numbers are increasing. So just hang tight. Let me share my screen while people are waiting. Feel free to go to this website. I'll show this again in a few seconds, but you can scan this QR code with your phone, or you can type this URL in your browser. It's case sensitive, so make sure you use the capital X and capital R when you write the URL. And this is just a basic survey to find out how many people use which of our resources. And I'll show you the results here. So let me go ahead and start this. So reload this page. Okay. All right, let me check one thing. I think somehow this did not reset since last time. Let me do, sorry everybody if you went ahead, and I'm gonna go ahead and stop this. Something went wrong with the resetting. It was still collecting information from last time. So I'm gonna ask everybody to hang tight and redo the survey if you've done it. Give me a second here. Let me reset the results. Okay, and all right, so please go ahead and do this one more time. I apologize about this. And I'll show you the QR code again. So go ahead and scan this or type the URL in. And the URL is also shown here at the top of the page, so you can type this in. All right, we'll get more accurate numbers about who's actually in this webinar. I'll just show the QR code one more time. So if people still want to scan, I'll give you a few seconds and I'll go back to the live polling. And I'm going to keep this poll open during the presentation, and we'll come back to it later on as well, and I'll show you the link. So if you didn't vote yet, and or you didn't get a chance to do that, you can do it towards the end as well, or anytime you feel like it throughout the webinar. Some basic logistics before we get started. So in GoToWebinar, in your control panel, you will you'll notice there's a section where you can ask questions. So throughout the webinar, feel free to type your questions if something is not clear or you have additional questions. If we can answer it by chatting with you, we'll do that. If not, we may answer it verbally, and if it's an answer that we don't know or a question we don't know the answer to, we will follow up with you in an email afterwards. Okay, the other thing is this meeting is being recorded. So if you miss anything, you'll get a chance to watch this again afterwards. And of course, you can share it with your colleagues if you think other people may be interested in this webinar. Okay, so in case you didn't know, today's webinar is going to focus on running an RNA-Seq and Galaxy workflow in VEuPathDB and integrating your data back into VEuPathDB. And I'll cover different aspects of running a workflow, and we're going to be demoing a couple of workflows, but one of them is just a very simple workflow. We're going to build it together, and then the second one is going to be a more complicated RNA sequence analysis workflow, and we'll go through that as well. If you're interested in all of our webinars, so we're adding weekly webinars, at least for the foreseeable future. So the URL that's on that page, which we will, somebody from the group will paste it in the chat window as well, will take you to this page, and you'll see here there are a number of scheduled webinars. Today we're doing the Galaxy one. Next week it's manual gene annotation in Apollo. Following that, we'll be doing Go, gene ontology, and enrichment analyses. After that, we'll be talking about motif searches and regular expressions. So if you're interested in interpro domains and how to find genes based on their presence of PFAM domains or interpro domains, but also a mechanism for you to define your own domain using something called a regular expression. That's actually, but the tools are all web-based, so anybody can do them. And then the final webinar, or the last webinar that's scheduled so far, we'll be scheduling more, of course, is on June 11th, and that will be covering our new tool called MapView, which allows you to visualize and explore population level data, both from vectors and parasites and so forth. So in today's webinar, I'll be doing most of the presentation today. I'm Omar Harb, and I'm based at the University of Pennsylvania. I'm joined today by a number of people from the outreach team, and those include Evelina Bashenko, who's based in Liverpool, Gloria Giraldo Calderon, who's based in Colombia, and Suzanne Warrenfeldt, who's based at the University of Georgia. And all of them will be helping answering questions throughout the webinar, and they may interject during the webinar if they want to clarify something or they have additional questions and so forth. Also, you may have noticed that there are 3 handouts available to you for this webinar. One of them is a general handout on how to access our Galaxy instance, and then the other ones are more detailed exercises that are based on the RNA sequence tutorial that I will, example that I will run through today. So you're obviously, those are things that you can take and try them on your own. They're very detailed and have a lot of information in them, and I would encourage you to explore them as well. Okay, so a couple of things I wanted to cover. So in Galaxy, so actually let me go back here. I'm going to just show you Plasmodium. This is one of our sites. The VEuPathDB resources have about 10 different or 11 different resources, including Plasmodium, which you're showing here, but VectorBase as well, which we've recently merged with VectorBase, and soon we'll be releasing new sites that are that will integrate all of our databases under the similar infrastructure and similar tools and stuff like that. So that's an exciting development. But today what I want to describe is this ability to analyze your own experiment. So any of our websites has a section here called Analyze My Experiment. And when you click on this, this will take you to a Galaxy site, which will look like this once you go through the click-throughs. And the handout, the first handout that is available to you describes this whole setting up your VEuPathDB Galaxy account. Basically, the same username that you use for logging into VEuPathDB sites will be the same username you use to get to Galaxy. But this is we've done it in a way that's automatic. So when you click on Analyze My Experiments, you'll get a bunch of windows, and these are screenshots here, which will walk you through the process of how to log into your Galaxy account. So and Galaxy is a really cool web-based interface that allows you to run various bioinformatic tools and software without knowing how to code, right? And so there's no reason for you to run Unix commands and launch software command line. You do it all using this web interface. So Galaxy was developed by we didn't develop Galaxy, of course. We're more than happy to take advantage of this amazing tool. And if you go to usegalaxy.org, this will take you to the main resource. You'll notice here it looks a little bit different from the version that we have on VEuPathDB sites, but that's because we're using an older version, which we'll be updating soon, and it'll look very similar to what you're seeing over here. All of these Galaxy resources, so no matter where they are installed, so in theory, your university or your group can install their own Galaxy instance, basically, and so you can make it available internally to your department. It requires somebody to actually do the work to maintain it and keep it going, but it's definitely doable. And I know many groups do this. They have their own Galaxy instances installed in their local institutions and they may install, custom software and tools in this left-hand panel. So this left-hand panel is basically a menu of all the different types of tools that are available to you. They're categorized options. For example, RNA-Seq, if you open this up, it'll show you all the different software that are available to you to run and explore. So the other amazing thing, so you can run single softwares, right? So you can click on any of these tools and say, well, I want to run a DSeq2, right? And you can click on this and you will get options. You can basically start running it on data you have uploaded into your Galaxy account. But a really cool thing about Galaxy as well is that you can build workflows, right? You can tether tools with each other. And so I'm going to take you back here and take a look at this. So I think most of the people on this call, I'm assuming, and based on the email list, most people on this call are people who are used to working in the lab, working in the wet lab. And as soon as, at least from experience, as soon as you start talking to people about workflows and software and putting them together, it sort of becomes very complicated and hard to visualize. But my background is a wet lab biologist. I've worked in a lab many years, and a workflow is really pretty straightforward. We run workflows when you're in the lab all the time. And what I'm showing you here is a very basic, simple workflow. It's a extraction of DNA from a gel slice. So let's say you've done a DNA digest and you ran a gel, and now you want to purify a particular band of your molecular size. So if you've already done a workflow before that, just simply running the gel. But this here shows you the workflow of purifying the DNA from a gel. You're coming in with an input, right, which is your gel slice, and then you're manipulating it. So it's becoming an output for this step here, where you add binding buffer, and then you incubate it, and then you add it to a column, and you spin, and you wash it, you spin again, you wash it, then you elute, you wash it, and then you come out with another output here. And so pretty much what you're doing is, let's see if we can do it here. There we go. So pretty much what you're doing is a bunch of steps, right? And each of them have an input and an output. Okay. So this is a workflow. You're manually doing it, right? So each step you have to go in and spin it, and then you put it in the incubator and you wait, and then you add another buffer and you spin it, or you do something else to it. But nevertheless, it's a workflow. You have inputs and outputs, and things are happening in between. So, and this is an example of a wet lab workflow. A dry lab workflow is basically the same thing, but instead of working with, physically working with DNA or material and manipulating by hand and pipetting and adding buffers, you're doing all of this virtually using different softwares. And so this is a very scaled down RNA sequence analysis workflow. So you come in with this file called a FASTQ file, which is basically a text file, a structured text file with a bunch of sequence in it. I think everybody on this call probably is familiar with what a FASTA file or a Fasta file is. It's a structured sequence file, and so a FASTQ is very similar. It's a structured sequence file coming from your sequencer, and then you do different things to it. So you run a software called, let's say, Trimmermatic, which is going to trim out the bad reads and low quality scores and so forth. So your input is a FASTQ file. You do something to it. The output is a FASTQ file, which is the input for the next step, which is in this case something called HiSat2. And again, don't worry too much about these different programs. These are things you will learn from reading papers, right? You look in the methods section, just like if you were going to try and use a new method for DNA purification, you might go to the paper and look at the methods and say, well, what did it do? What was their workflow? They used this buffer, then they incubated, then they did another buffer, and so forth. And this is the same thing. You would look in the methods and you'd say, well, what tools did they use? And then they will explain, oh, we used that tool called HiSat to align our sequences to our genomic file and our genomic sequence. And so you run this tool. Now the output, you'll notice it changed. So it started with a FASTQ file, now it's something called a BAM file. So just like in the lab, you might start off with a DNA in a gel slice, right? And now the next step you have now, instead of a gel slice, it's a solution of gel and DNA, and then in the end, you have just DNA, right? And so you can imagine different inputs, or inputs will change and result in different outputs depending on how you manipulate them. This BAM file now is an output from the HiSat step, but it's also an input to the next step, which is Htc Count, which is going to count the number of reads per gene, let's say. And so now what you come up with next is a table, basically, with a count file. So each gene with the number of reads that align to that particular gene. And then you may want to run, if this was an experiment with replicates and you're comparing samples to each other, you may want to run something called DESeq2, which looks at differential expression. And again, the output is another table. So there's still tables, but it's another table giving you some statistics and telling you something about how confident you should be with the differential expression. Okay, so let's go back here to use Galaxy. So I'm going to demo a very basic workflow in use Galaxy, and then we will go back to our Galaxy in VEuPathDB and demo a more complicated one, and then take you back to VEuPathDB and show you how you can work with the data that you've analyzed. So let's start off by saying we're going to create a workflow. So let's talk about, well, what does this workflow look like? So I'm going to go to the workflow section right here, and this will load all my previous workflows, which is fine, but I'm going to create a new one right here. So I'm going to click on create. And so what this, gives you an option here. What do you want to call it? So let's say we want to create a workflow where I want to align a number of amino acid sequences, because I want to ultimately generate a tree from this alignment. So let's go ahead and call this whatever, my alignment. Save it. Okay, so now you have this empty workspace right here. There's nothing in here. It's like a little canvas. And now the question is, well, how do you build a workflow? So we're going to have to use tools from this left-hand side and start our workflow. So the first thing we want is our input, right? We have some input data, right? And so we have to tell the workflow that we need an input. So I'm going to click on inputs, and I'm going to say it's an input data set. And now you click on this and you get this little box, okay? This is going to represent my sequence file of amino acid sequences that I want to do an alignment on. The next thing we need to do is we want to find an alignment program. So Galaxy has this really nice filtering tool or filtering option at the top here. So I can just start typing alignment, for example. And depending on how things are categorized, you look in here, you'll see there are many different alignments. So sometimes it's depending on how many alignment tools are there, it's really easy. You will find it right away. And other times you kind of have to sort through a little bit more. You might have to do it differently, but here you'll notice this is a category called multiple alignments. So this is probably what I want. I have multiple amino acid sequences that I want to align. Here are different algorithm or different tools, software that actually do alignments. ClustalW, I think, is something that many people are familiar with. So I'll go ahead and select ClustalW. So once you click on this, it generates another box for you here. This is the ClustalW program that you're going to run on these sequences, okay? So, but ClustalW doesn't know what it's going to run on, all right? You have to give it the sequences. So I'm going to click on this little arrow, and I'm going to connect these to each other. Okay, so I've just made my first workflow, right? I'm starting with an input sequence, and I'm going to take my input sequence and send it to ClustalW. Of course, your input sequence has to be in a particular format, right? In this case, it's telling me that it needs to be a Fasta file, okay? It's just like in the lab, if you're trying to purify DNA and you took you took a bunch of protein, a sample of proteins, right? That doesn't have any DNA in it, and you try to purify DNA from it, it's not gonna work, right? And so same thing here. The program will require the input file to be of a particular format. Okay, and then let's say the next thing you want to draw a tree based on this alignment. So we can do. I'm just gonna type another filter, tree. And let's try and look and see what options are available. So there's this Newick display, visualize a phylogenetic tree. That looks interesting. So I'm going to go ahead and select this. Okay, and let's go here. I'm just going to move it here just so it's easier for us to look at. So I'm starting with my data set that's going to go into a cluster W. The cluster W requires a Fasta file, and you'll notice here there's some output formats that it's going to generate for you. It's going to generate this file here as a filep or Fasta, and also it'll create this other output, a DND or an HX format. Okay, don't worry about the formats. The point is that you look at these two things here and ask, well, do I have the right format to go into this program? So once you click on any of these programs, you'll notice here on the right, there's a lot of information about the program. So I can go down here and look, and it tells you what input. So the input needs to be in any of these formats, and you notice here NHX is one of them, and that's one of the outputs. So that's great. So I'm going to take this output and connect it right here. Okay, so here's my first workflow. It needs an input sequence. It needs it's going to generate a cluster W is going to run on these sequences, and then it's going to draw the sequences, and then I can save this. Okay. So the next thing we need is we need some data. All right, and so to get data into Galaxy, there are many different ways. Let's go ahead and delete this over. But you'll notice here there's a get data option, and so when you expand this, there are many different ways to get data into Galaxy, which is really cool. In my case, I just want to upload it from my computer. So we can go ahead and actually go back to my Galaxy view right here. I'm just going back to the home page right here. And I've already done this. I've uploaded the data set from my computer, but if you click on get data, and let's go down to so somewhere in here, which I'm not seeing it right now for some reason, there's an option to upload data from your computer. But the other easy way to do it as well is to click on this little link on the upper side right here, which basically is a shortcut to the same option. You'll get a pop-up here, and now I can choose a file from my laptop. And in my portion here, I already have a file that's a Fasta file that has multiple amino acid sequences. I can choose this, and I can start this process. And so what you'll notice is that it says it did it. I close it, or it started the process. I've already uploaded the file, so it's green right here, but you'll notice this gray one here is the process of the file being processed and uploading. Okay, so this takes a few minutes, but I'm not going to wait for it. We'll let it go in the background. But since I already uploaded my sequence right here, and we can take a look at it by clicking on this eye, you'll notice here it's a multi-Fasta file. So hopefully everybody's familiar with this. There's the name of the sequence, or the description, and then the sequence itself, followed by the next one. And I want to do a multiple sequence alignment on this. And we already prepared a workflow to do this. So I'm going to go ahead to my workflows. And this is my alignment. This is the one that I just generated. So I could either click on it and ask to run it, or right here, I can click on this little arrow to run this workflow. Okay. So once you click on run, it gives you these options. So you'll notice here that my input, it's looking for a Fasta file. There's only one. It's called everything.fasta. And as I was doing that, you notice that it already uploaded the new sequence, basically, which is fine. I can use either one of them, basically. Cluster W, if you open this up, you'll notice here there are a bunch of options. So you can potentially play around with them. The drawing file, you notice there will be a bunch of other options, including the output format. And so in my case, I want to change the output format from this SVG, the support vector graphic, to a PNG. I want to be able to visualize it right here. Okay, and that's pretty much it. There are a number of other things you could play around with if you want, but that's it. So then you click on Run Workflow, and what's going to happen? It's going to tell me that there are a bunch of steps that are scheduled, and it'll give you an idea of the status of everything. And once they're running, you will notice the portion here will start populating with the outputs of your different tools. So rather than waiting for this to happen, because it'll take a few minutes, I've already pre-run this, so I'm just going to switch my workflows. You can have multiple different histories. So these are, imagine them as little folders with different experiments in them. And so I'm going to switch to a history that I ran earlier today with that alignment. And so I'm just going to click on View All Histories and wait for it to load. So sometimes you have to give it a little refresh. Okay, so these are all my histories. This is the one I ran earlier, so I will switch to this history. You'll notice I have additional histories here. Some of them have red boxes on them. That indicates a step that failed. So just like in your lab workflow, you run your gel and you say, oh my God, there's no DNA in there, something didn't work. You'll have to go back and try and troubleshoot the different steps. Well, why did this not work? Well, did I have the right input? Did I have the right buffers? Did I have the right temperature and so forth? So same thing with this, you'll have to go back and play around with this. So I'm going to switch to this workflow from earlier today. All right, and now it puts it here in the front. And now let's go ahead back to the home page, and we will see this workflow now the completed workflow will show up here. And I ran this in two different ways. I ran it as a support vector, but you'll notice here now, this is my everything Fasta file. These are my clustal outputs, and you could visualize them and view them. So this is, for example, this NHX format looks like this. And so the example is down here, but you can load the whole thing right here. And this is the format that will be used by a tree drawing tool, which we have one, the Newark display data tree. And so if I open this one, you'll notice it's in a PNG format. I can actually look at it right here. So I'm just going to click on view data, and here's my tree. Okay, and I had selected that I wanted it to be a radial tree, but you can actually change this to be a regular dendrogram. Okay, so hopefully this was sort of a very short primer on how to build a simple workflow and run it. The Use Galaxy site has a large number of online resources to teach you how to run different types of workflows. It's really an incredible resource, and we have a link to it in one of the handouts. And I would really encourage you, if you're planning on running workflows, to go through these modules. They're really guided learning modules, and they're really superb. I've used them. I learn a lot from them every time I try them out, and so that's a pretty cool resource. Okay, so I'm going to go back to Plasmodb, and so we've installed our own Galaxy instance, and the reason for that is twofold. One is we wanted to be able to preload all of our genomes. And so you don't have to worry about loading the genome or figuring out, do I have the right version of the genome that's the same in the VEuPathDB databases. So we preload all the genomes, and they're available for you for any tool that requires the genome and the annotation. And the second thing is that we've built in some tools that allow you to then transfer analyzed data back into VEuPathDB. And we're building many more tools coming hopefully in the next few months that will allow you to transfer different sorts of data. But once you click on Analyze My Experiment, you might have to click through a couple of things, but it'll ultimately take you to a page that looks like this. So this is an older version of Galaxy, but it's the same idea. You have a panel on the left with all the tools, a middle section here, which is basically where you visualize your data or where you build your workflows, and then a right-hand section here, which is your history. Okay, and what I'm going to do for the next few minutes, probably the rest of the webinar, is I'm going to take you through the process of running an RNA sequence analysis workflow. We're not going to build the workflow. It will be a very complex workflow. It will take us a whole hour just to build the workflow. So rather than doing that, we have already some pre-configured workflows, and you'll notice here on the home page we have some example workflows that we suggest that you start off with. But any of these workflows can be modified. So as soon as you click on any of those, it gets imported into your own workflow space, and it becomes yours basically. And you can modify it, or you can run it as is, depending on what your goals are. And so I'll show you what this workflow looks like. So I'm going to go to the workflows. This is all the workflows in my space. You won't see this if you go to your workflow, but I'm going to go ahead and look at this one here. I'm going to edit it just to show you what it looks like. So remember, we built a workflow in Use Galaxy, which is a very simple workflow. In this case, this is a slightly more complex, but really in the end, it's really straightforward, right? You're starting with input data, and you're going into tools which will generate more output, different outputs that you take into other tools. And in my case, this is a particular workflow that takes two collections. A collection is basically a way for you to group samples that belong to each other into one group. So I'm going to be taking samples from 2 different developmental stages in Plasmodium falciparum. I'm going to take, I think I'm going to compare erythrocytes, asexual stages to male gametocytes. And so one collection will be my erythrocyte samples, and I did this in triplicate, or the people who did the experiment did it in triplicate. I'm just going to use their data. And so there'll be 3 samples for, or 3 replicates for the erythrocytes and 3 replicates for the male stages. And so I have to tell it I have 2 collection inputs, and then each one of these is going to go into its own workflow before they get joined together to look at differential expression. So there's a tool called, I'm not going to go through the details of each of these tools. These are described much better in different places, but essentially we'll go through some processing of the file. FastQ groomer makes sure it's the right format and everything. FastQC will check the quality of your read. So this is something nice for you to look at and see, well, did my sequencing work? Are there any red flags that I need to be concerned about? Trimmomatic, so your output files will go into a program called Trimmomatic, which is going to trim all the bad reads. HiSat2 is going to align your output from Trimmomatic to a genome. Then the output from HiSat2 is going to go into 2 different things. The output is a BAM file, which one, it's going to be converted into a bigwig file. It's just another format for visualization. And also this file is going to be used to generate the counts. Once you have the counts, they will be combined back together into DESeq2, so you can determine if there are any differentially expressed genes between these 2 groups of samples. Okay, so this is the workflow that we're going to run. So let me go back to the home page. Yes?
I just would like to add one more thing. If you could navigate back to the edit screen. If you click on individual steps and highlight essentially tools that are used to create a workflow, the panel on the right will change and expose additional information such as how to access manuals and also the specific parameters that are important to keep in mind if you want to get your settings customized even further from the default settings.
Yeah, that's a really good point, Eve. Thanks for pointing it out. So, and I'm showing you here, I clicked on DESeq2, for example. There's a lot of information here that's specific to this version of DESeq2 that's installed, and lots of parameters you can change. And the default ones that we have may not be the ones that you want. So just like in the lab, when you're going to purify DNA, you may be working with an organism that has a high AT content, and then the standard DNA purification may not work. And so you're going to have to modify your buffers, maybe use a different binding agent or something to improve your purification. Or if you're designing PCR primers, same thing. Depending on what your target DNA is, you may have to modify the primers. So this is the same thing here. Each of these steps you can custom them or tailor them to your needs, and also there's a lot of information about the tool itself, including links to where this tool came from and the manuals, and so there's a lot of information in here. And I would recommend, obviously, if you're doing this experiment yourself, to take your time and really understand each of the steps and determine if these are the steps that you really should be running in yours. You may decide, well, I don't like Trimmomatic. I want to use a different tool for trimming. And so you can look and you'll find that there are other tools for trimming in here in the database. And so you may decide I want to use this FastQ trimmer or this program called Sickle, right? And so you can click on this, it'll add it to this workflow, and then you can start, you can actually change this workflow to for yourself, basically, depending on what you need. Okay. All right, so the next step is you want to get some data. And so there are different ways of getting data into a Galaxy instance. In this case, so if you had a large file on your computer, the instance we have uses Globus data transfer, and this is a high-speed way of transferring data. I won't go through the details of how to set this up because there's actually a lot of very useful help on how to create your own Globus Personal Connect endpoint. So that's something that you'll install on your own computer, and then it'll allow for fast transfer of data from your computer or from a server to your particular workspace in our Galaxy instance. in my case, I'm going to get data via Globus from EBI. So I want to get the data from the EBI server. So I'm going to click on this get data via Globus from the EBI server. And if you look at one of the handouts that was provided, the RNA-Seq part 1, this will lay out the experiments I'm interested in. So I'm looking at erythrocytes and males. These are the ones I want to compare to each other. So I'm just going to demo this for one of the samples. I'm going to take one of these sample IDs. This came from the Sequence Read archive or the European Nucleotide Archive, which mirror each other. And let's go back to our Galaxy. I'm going to put the SAM ID right here. I know when you look at the details of the experiment, they told me that this is a paired end experiment. So for each sequencing run, you'll have 2 files, basically, one for the forward reads, one for the reverse reads, because each fragment was sequenced from both ends. And then I can click on execute. And once you click on execute, it's going to load. It shows you what's happening here on the left. You'll notice here that there are 2 files, one for forward, one for reverse. So each sample is going to give me 2 files. And for some reason, this failed right now, but sometimes you just have to reload it and try it again. And it should work because I did try it a bit earlier and it was working. So we'll just let this go and in the background. So I did to make sure that we don't spend all the time on watching this work. So you'll notice now it's working this time. Sometimes it fails for there's probably a bad connection or something. So you just have to try it again. It turns yellow when it's running and then green when it's done running. And so I'm going to go ahead and take a look at. Let's see. Go here. So I'm just clicking between different histories that I have, and you notice here there are 12. It should be 12 files. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Yeah. So there are 12 files in here, right? So we had 2 experiments, erythrocytes and male gametocytes, and each one was in triplicate. So I have 3 samples, and each sample was sequenced for both ends. So each sample provides 2 files. So 6 times 2 gives me 12. Okay. So these go together, and then these go together. So now I need to define this. I need to put these into what are called collections, into these folders. So I want to group these 6 together and then these 6 together. And so I'm going to show you how to do this for one of them, and then we'll move on to the next stage. Well, actually, I'll do both of them very quickly. So what I'm going to do is I'm going to click on this operation on multiple data sets option right here, and I'm going to select the first 6. Okay. And now for these selected, I'm going to make them into a data set of pairs, okay? Or a list of data set pairs. There we go. Oops, sorry. A list of data set pairs. That's what I want to select. Okay. So it automatically pairs them together based on the name, and you'll notice here the same ID _1, the same ID _2, because those belong together. And I can rename this quickly. I can call this E1 for erythrocyte 1. E2, E3, okay? And I'm going to call these erythrocyte, create list. Okay, so now you'll notice here there's erythrocyte, and these are all still checked. What I'm going to do for selected, I'm just going to hide those selected because I'm done with them basically, so they're going to hide. And now what I'm going to do is I'm going to select the next 6. Same thing, build a list of data set pairs. These are the males, so I'm going to call them M1, M2, M3, and we'll call these males and create list or collection. So now you'll notice here there's a male collection, and that same thing I'm going to hide the ones that I'm done with. Okay, they're not. This is obviously they're not being deleted. They're just hidden. This makes your workspace a little bit cleaner. And now if I click on this, erythrocytes, for example, you notice there's E1, it's a pair of data sets, E2, it's a pair of data sets because they're pairs forward and reverse, okay? So I have now a collection of my erythrocyte samples and a collection of my male samples right here on the left, on the right. So now the next thing is I'm gonna run the workflow on it, right? So I'm gonna go ahead to my workflows. You can either do it, actually, let's go back here and let's go to the home page because that's probably most likely how you will run it. So once you have your collection set up, you can go back to the home page and I can select from these workflows. I'm going to select a workflow for paired and unstranded. My particular experiment was unstranded, but if you had a stranded experiment, you can actually run it in the stranded using the stranded options. So they're just the same workflow, just pre-configured for stranded RNA sequence data. And so once you click on this, this will actually immediately import this into your workspace and initiate the option to run this experiment. Okay. And so let me go back. I think I might have selected the wrong one. Let me go back here. Let's see. Oh, sorry. Yeah, I selected. So I have to read carefully what I'm picking. So I selected one. I want to do differential expression. So I'm going to pick the same workflow, but this one allows for differential expression. Sorry about that. So, but again, the bottom line is that you want to make sure you're selecting the right workflow for the type of data you have. I have 2 samples. The previous one I loaded only had one collection available, so I knew immediately something was wrong. So I have my input collections. One of them is males, and one of them I'm going to have to change it to erythrocytes, right? Because I want to compare these to each other. As you go down, you'll notice all the steps that I showed you in the workflow are available for you here, and many of them may have options that you want to change or not, depending on what your needs are. So in my case, I'm going to keep almost everything at the default. So FastQC, traumatic, I think those are all defaults are fine. HiSat2 is the step that's going to align your sequences to the reference genome. So you'll notice the reference genome selected by default is the one that starts with A, and it's the first one in the VEuPathDB. We don't want this, right? We don't want to align the malaria, the Plasmodium RNA-Seq data to an amoeba genome. It won't work. So let's go ahead. And in this particular case, it was Plasmodium berghei. So I know this from the experimental details. So I'm just going to pick just shortcut to Plasmodb. And as you scroll down here, you'll notice that one of them is Plasmodium berghei. So I'll go ahead and select this. And because you have 2 different groups, you're going to notice that you'll have to do this twice, one for the male, one for the female. So again, I'm going to pick Plasmodium, and I want to pick Plasmodium berghei. Okay. And as we scroll down now, you'll notice now you have this Htc Count, and this also requires the annotation file. So I need to tell it what genome it is. So I'm going to pick Plasmodium again. The same, one of the most important things here is to make sure you're selecting the same reference genome for all of the steps that are in the same workflow. Another thing you'll notice here that it's going to create counts on features that are in your annotation file, right? So you have to tell it everything. It won't know this. By default, it's selected as exons, but I want the counts to be on the genes, not the exons. So I'm just going to change this to gene. Okay. And let's go ahead down to the next Htc option. And again, I'm going to select Plasmodium berghei. There it is. And let's change this to any other options to change here. Feature type, we changed it to gene, so let's change this to gene. Okay. And that's it. So these are all the things you have to change in this particular workflow. You can hard code these changes. So if I go to the workflow itself, if let's say you only work on Plasmodium berghei and you're always going to be using the Plasmodium berghei reference genome, and you always want to create your counts on genes, not on exons, then you can actually change the workflow itself and save it and say, this is your modified workflow. And then next time you don't even have to make any of those changes. You just run it. And so I'm going to go ahead and run this. And so now what's going to happen as this starts to run, it's going to queue up all the steps for your workflow. And you'll notice my right-hand panel will start populating with many, many files. So ultimately, it'll generate over 100 different files that are going to be inputs and outputs for each of the steps. And this will take a few hours to run, right? This won't happen instantaneously, okay? So I did obviously create one that's done. So I'm going to go ahead. So you'll notice here in our workflows, the one that was called before collections, that's the one I was working on. You'll notice here there's some yellow steps. So these are running, 14 are still in the queue, and these numbers will change as it's updating. Okay. So let me go to this one here, which is are we? So this one right here. So this is what it'll look like once everything is run. So I still have my collection of erythrocyte and males. You'll notice here that they're the FASTQ options, FASTQ-C options, termomatic outputs, the HiSat outputs, and you can click on any of these and look at them, right? So for each one, there'll be, these are BAM files, right? And so you won't be able to view the BAM files here. They're too big. It's 1.1 gigabytes for each of these, so that's a lot of data. But you can certainly download them and view them in a browser. We do convert BAM files to BigWig files. This makes them smaller. So instead of 1.1 gigabytes, it's 34 megabytes. And this is easier to download and easier to visualize in a genome browser. Okay. So as we scroll up, you'll notice here there's the Htc Count. Okay. So these are your, if I click on this again, for each of the samples in your biological replicates, there'll be a table. And if I can view this, this has 5,245 lines. And you'll notice here, these are all gene IDs, and each gene has a value associated with it. This is the count of the gene, of the expression, basically, of this gene based on this particular biological replicate, E1, right? The same gene, let's say this one here with 404, will have a corresponding value in E2 and also a corresponding value in E3, okay? And the next step after you do your Htc counts, it's going to this program called DESeq2, which is going to try and determine if there's anything statistically significant differentially expressed between the males and the asexuals, the erythrocytes, right? And it's taking into account your biological replicates, of course. So that's where it's able to bring you the statistics of whether this is something you can trust or believe or not. Okay, and so this DESeq option will also end up generating a table. And if you view this, you'll notice there's a table. Each gene now gets multiple columns. Each of these columns, they're actually described. If you go to the tool itself, there's some description here about the columns, but also in the handout, we have a slightly more detailed description for the 7 columns that you get. But one of them will be your expression level, the fold change, and then also the statistics surrounding it. So this 9 times 10 to the minus 94, that's a pretty good statistic, and that this is potentially a down-regulated gene when I'm comparing the asexuals to the males, okay? So in the end, you can download these files. So any of these files, you can click on this download option here. And when you click on this, it's gonna just download it to your computer, and you can open it up in Excel. So often, you know, there's nothing magical in the end. In the end, you come up with tables and potentially some graphics, but then you have to manipulate them further in other programs. And depending on your expertise, you may end up just using Excel, which is totally fine. Or you may end up using other programs that will parse out interesting things from these files, depending on what you're interested in. So notice here in your output, it says 26 shown, 109 hidden. Okay, so there are 109 files that are hidden from view. Those are different outputs, including the ones that I hid earlier. Remember when I created these collections, I hid all the individual files. So those are all part of this hidden file, the hidden files. So one thing I want to do is I want to transfer my counts, my Htc counts. I want to transfer them back into VEuPathDB so I can actually ask questions about them within, let's say, Plasmodium. And so the exercise, and so the way to do this, at least right now, we allow you to transfer back FPKM values, okay? So FPKM is one type of normalization. It's not the favorite normalization these days, and so we're going to be changing to counts per billion. So that will change. But for now, it is the option to you have to get FPKM values, and there are different ways of getting these FPKM values. And so what we're going to have to do to actually get this to happen, because none of the steps in my workflow up till now, up here are some steps that I ran afterwards, which I'll show you how to do this in a second, but up till now, up till here, none of these steps generated FPKM values. Okay, so you'll have to find another tool to generate FPKM values. The exercise that was that's provided to you will walk you through the process of running a tool called Cufflinks, which is available here. and cufflinks will then allow you to generate FPKM values. We have another tool called under ViewPathDB RNA sequence tools called Htc Count to FPKM. So I can go directly from the counts to FPKM values using this tool. And that's the one I'm going to demo right now. So, but before you do that, there's a little bit of preparation work you have to do. So one thing I need to do is I need to put all my male counts and all my asexual counts into one collection, okay? And so this may sound a little bit tricky here, but I'll just walk through the process mechanistically so you can actually see what it looks like. And so the first thing I'm going to do is I'm going to show all the hidden files. So I'm going to click on hidden. Okay. And now you'll notice that there are, as I scroll down here, you'll notice that there are many, many, many files that are available to you. So I'm going to start from here, from this DE-seq2. Anything above here is new stuff that we're going to replicate. But as I scroll down here, you'll notice that there's Htc Count. Each Htc Count step generates two output files. One is called Htc Count on data, no features, and one is on data. The one we want is the one without the no features option. No features, it means these are reads that did not map to any of the features that I described, so did not map to genes. So I don't really care about these for now. So for now, I'm going to care about these Htc Count on data. And you'll notice there, if you think about it, you will have, you should have three, for each of your paired end files, you should have one of these, right? So if you open this up, you will get a count for reads. So each one of these corresponds to the E1, E2, or E3, or M1, M2, M3 in the Htc Count collection, okay? So in the end, I'm going to create a collection that contains 6 Htc files. Each three of them are from one of the conditions. And so let's go ahead and quickly do this. So I'm just going to click on this little check mark, and now I'm going to pick these Htc Count ones. So this 102, 101, and 100. And now let's scroll down. Oh, sorry, I'm picking the wrong feature. No feature, there's another one. So I'm going to pick all the ones that say that do not have the no feature option in them. Okay. So here are these three, and I'm going to scroll down a little bit more. Should have some more somewhere else. So any Htc Count that doesn't have no features on it, that's what I'm going to select. So here's another one. This is a collection. Here's this one, and this one. Okay. And now for selected, I'm going to pick, build a list of dataset pairs. Or it might be build a dataset list. So let's find out which one it is. So that's one option. Sometimes I have to click these, and even if I ran it right before the call, I have to pick them again and make sure I'm running the right one. So this is not the right one. So let's cancel this. So I think it is build cancel. And let's pick. So I didn't pick something correctly here. Okay, yeah, so this is what it is. So basically, remember I'm creating one collection, and these three are, I know this because they're in sequence, so these three are going to be my erythrocytes, and then these three are going to be my males. So the one quick thing I'm going to do is I'm going to just change these names to E1 again, E2, and E3, and this is going to be M1, M2. And so you'll notice that as you're working on these, it's almost like working in the lab. You're going to have a lab book or somewhere where you keep notes. There's a lot of files that are being generated, and it may seem overwhelming, but in reality, it's not more complicated than running an experiment in the wet lab where you have to keep notes and keep track of what you're doing and understanding each of the steps. So a lot of it is obviously trial and error, but let's call these males/females. Sorry, not females, this is asexual, so and asexuals, and create list. Okay, so now it created this data set list of males and asexuals. Okay, And now the next step, I'm going to run this Htc Count to FPKM. I'm going to pick a collection, and I'm going to take these males and asexuals. It is not stranded. I need to tell it which annotation I want to choose. So in this case, again, we're going to pick the same Plasmodium berghei annotation, and I'm going to execute. Okay, so this is going to now the same thing. It's going to run a bunch of steps, and you'll see a bunch of things happening here. We're not going to wait for them, obviously, because I pre-ran these. And so what happens, you will notice here, you'll get a Htc Count to FPKM on collection, and there'll be two of them, one for antisense, which in our case we don't care about because this wasn't a strand-specific one, and then there's one for gene expression. So if you open this one here, you'll notice that there are values here for each one, and for each gene ID now you have an FPKM value. Okay, so this is exactly the file that we need so we can export to VEuPathDB. So now the next step is I'm going to use one of these export tools. So I'm going to open up VEuPathDB export, and I want to export RNA-Seq to VEuPathDB. So I'm going to go ahead and select this. And now we're going to have to give it a name. So let's call it my best experiment. And is it strand specific? It is not. Bigwig, so we can go ahead and do the same thing with the Bigwig files, put them into one collection. I didn't do that for the demonstration here, so I'm just going to pick one of the collections. So it'll be probably the male ones, which is fine. If you put them all into one collection, you'll be able to transfer both the males and the females. But what we're interested in is this FPKM collection, and so I'm going to select the FPKM collection here on genes, and I believe it's 136. Let's confirm that we got the right one. Yeah, it looks fine. These steps even already ran. So let's go ahead and select this. And I'm just going to copy my best experiment into the other fields because I'm not going to worry about providing a good summary and description right now. And then you go ahead and click on execute. Okay, so this is going to transfer this data now to VEuPathDB. It may take a few seconds. We won't wait for it. I already transferred it earlier today. And so let's go ahead back to Plasmodb. So now in Plasmodb, I'm going to go to my data sets right here. And if you click on this, it's going to open up a list of data sets that I have in my workspace. So there are many data sets here, but the one that I transferred earlier today, it's asexuals and males. You know, in a few minutes, when I reload this page, there will be another data set here based on called my best experiment. So whenever that part is done, it'll show up here, and I'll come back to it later on. We'll see if it worked. So I'm going to go ahead and click on asexuals and males, and you have your own private. This is all under my account, so you can log in and create an account. And now I have my own private space for my data set. And you'll notice here it has information about the data set, but also tells you what available searches are there, and also it allows you to view your samples in a browser. So if I click on View in Genome Browser, this is going to take one of my Bigwig files, which is a representation of the coverage. It's going to open up the genome browser in Plasmodb for Plasmodium berghei, and it's going to load that specific Bigwig file. And so you can look at it and explore the actual coverage. And if you're interested more about JBrowse, we have a couple of webinars that we ran a few weeks ago. Those are all available as recordings. So you can view these and you'll learn more about how to navigate JBrowse. And then the really cool thing here is this available searches RNA-Seq user data set. Remember, we transferred the FPKM values. So now we can go ahead and click on this step here. And now you get a search page. And this is, again, it's a private search page just for you, for your data set. So I can say, well, I'm interested from my experiment. I want to find all genes that are upregulated in males. So let's pick all the male samples here as in the comparison, and I'm going to compare them to my reference, which will be the erythrocytes in this case. And I could play around with these options here. So this tells me that I'm looking for genes that were upregulated by twofold in my males right here compared to my females. And a twofold has to be between the minimum expressor of the 3 replicates and the maximum expressor of the 3 other replicates. Okay, I can change this fold change. I could change it to 5, let's say. So I'm looking for something that's a bit more differential or upregulated. So you get 5-fold in this case. And now I can click on Get Answer. And once you do this now, I get a step here. And if you're curious about these in silico steps or strategies, we had a webinar last week, which is also recorded and you can watch. It provides you a detailed description of how to run a search strategy. So this is a step in my search strategy, but these are 314 genes from my experiment that were upregulated in male gametocytes compared to the asexual stages. And you can scroll down and look at these genes. And if you go here to the right, there's a custom graph just for you that's built on your data, shows you the male samples and the erythrocyte samples. And so you can confirm, oh yeah, this looks pretty good, okay? This is pretty cool because now you can start asking questions about your own data sets from your own experiment that are differentially regulated. And so in this case, there are 314 genes. I may ask, well, is there something interesting in here? So I could potentially analyze my results and run a Go enrichment. And if you're interested more in Go enrichments, I won't go through the details right now because in a couple of weeks, we have another webinar that's all about gene ontology and different types of enrichment analyses. But suffice it to say is that you can take any list of genes, and in this case, these are genes that came from my experiment that are upregulated in males, and you can run an analysis on them, and you'll notice here that the cytoskeletal part and so forth are pretty, they're decent p-values associated with them. So that's okay. So then you can find some things that are potentially distinguished or functions that are important for your particular stage. And of course, you can add steps and say, well, how many of these genes are also secreted? So I can add a step here and look for protein targeting localization, and I can ask for predicted signal peptide. I'm just going to pick all, intersect. Again, view the webinar from last week if this is not familiar to you. And I can tell you now that there are 31 genes of my male upregulated Plasmodium genes are also potentially secreted. And now you can go and look at them a bit more in more detail. Okay, so we're at the top of the hour, and I know I've covered a lot of information here, but hopefully it gave you a flavor of what you can do both in Galaxy, in our Galaxy, but also in general in Galaxy in terms of running workflows. With this added functionality in the VEuPathDB Galaxy, the ability to transfer data back into VEuPathDB. And we're working on additional mechanisms so you can transfer the actual differentially expressed values, so you can actually use the statistics in the site. Also, we are working on mechanisms to transfer back variants. So if you did a SNP pipeline, for example, we're working on ways to do that. And so there are many more features coming available, ultimately, for you to be able to privately work with your data and analyze it further. So I'm going to, yes? Mm-hmm.
There was one question that I'm not sure that we answered, during the webinar, and one of it was, how are the fold change ratios defined from DE-seq? Right, so is it always sample 1 divided by sample 2? Do we have?
So in DE-seq, it's doing something a little bit more complicated, and there's some pretty good papers and resources out there that describe the math behind DE-seq 2. But ultimately, it's not only just comparing sample to sample, it's also taking into account the variability within the replicates of each sample.
Right.
And so, yeah, sorry.
I mean, we came up with 1 fold change ratio. So, I mean, I think the person who asked this question is correct. It had to have been sample 2 over 1 or sample 1 over 2. I don't recall how we set it up. I think it was males over erythrocytes.
Oh, you mean in the query that I ran here? Yes.
No, in the DE-seq results.
Okay. Yeah, so I guess I don't understand the question fully, but basically, whether we did it male versus whether it's the asexuals or vice versa, obviously, yes, it's going to do one over the other. You're right. So it depends on which one is factor 1 and which one is factor 2, at least in DE-seq.
Right. Okay.
Yeah. Mm-hmm. So I'll stay on for a few more minutes and see if there are any other questions that we want to answer. While we're doing that, I want to make sure that everybody has access to our survey. So this is our post-webinar survey, and your input is very valuable. I'll go ahead and copy this and put this in the chat box as well. Make sure that everybody has it. So yeah, so you can go ahead and either click on the link in the chat box or scan this QR code, and then you can fill the survey. Our experience is that if you fill the survey soon after the webinar, first of all, we get more people filling the survey, and second of all, things are still fresh in your mind. And so as the sooner you fill this up, the better. But I'll stay on for a few more minutes and see if there are any other questions, or maybe one more minute. One thing to note as we are waiting here, if there are any other questions, on all of our sites, we have a Contact Us link. And you'll see here at the top side or also at the bottom of the page. So make sure if you have any questions in the future, feel free to contact us. All of us on this call will be seeing those emails, and we're happy to answer them. We love hearing from people, and no question is simple enough or too complicated. Just send a question. If we know the answer, great. If we don't know it, we will tell you we don't know, and we will hopefully try and point you in the right direction. Great, and one more thing, again, just as a reminder, we do have additional webinars coming up. So next week, it's manual annotation and Apollo, but following that, it's Go, gene ontology and enrichment analyses. And then after that, we'll do motif searches and regular expressions. So, and then after that, we'll do map view. So a lot more coming, so feel free to sign up for those. Yeah, so somebody asked if I'm going to make the workflow for gene expression public. It is actually already public. So if you go to our Galaxy right here, and you just go to the home page. So if you click on Globus Genomics right here at the top, you'll notice these workflows right here in the center are all publicly available Galaxy workflows. So if you click on any of these, it'll import them into your workspace, and you'll be able to modify them and work with them. And you will also may notice that some tools you're interested in are not available here, and so you can always send us a request to install tools. And of course, maintaining these tools, because we're not running each of these tools every day, sometimes they go out of date, and you may try and run one and it won't work. Don't get frustrated and don't think that it's your fault. If something is just not working, just send us the information and we can help you. And you'd much rather hear somebody say, oh yeah, you should have changed this parameter, than you spending weeks on it trying to figure it out or feel like you can never do this. I think anybody on this call can run these types of tools. And just like an experiment in the lab, in the wet lab, they're complicated, but they're not, they're not magic, right? And so you can understand each of these tools. It just takes time, and you have to spend time on them to understand what you're doing and make sure you're running the right analyses. Okay, so with that, the webinar recording will be available to everybody. You'll get an email about this. We'll also post it on the webinar page. So all the previous webinars are, if you go to any of these, they're all posted on the bottom with links to the YouTube video of them. And so this one will be posted here as well. All right, thank you, everybody. And we will see you online soon again. Bye-bye.